All right, continuing on with the digestive system. So the movements through the stomach, so the rugae flatten, that allows the stomach to increase up to 20 times its actual size. So just by those, by the flattening of those folds, it allows that stomach to expand. Relaxation of the rugae mediate the reflex that's integrated there with the medulla oblongata. So it's kind of the trigger of that reflex. This is also going to inhibit muscle tone and pressure to prevent or minimize the ability of smooth muscle to stretch without increasing any of the tension. So again, allowing that stomach to expand. Now, as everything moves into the stomach, this is where we're going to get that mixing. So the mixing occurs in waves, in that peristaltic wave. So again, there's not just like continuous contraction, but everything is occurring in waves. So that works together to move the chyme toward that pyloric sphincter before it moves into the small intestine. And again, chyme is the bolus plus all the secretions from the stomach, like the mucus and the hydrochloric acid and all the enzymes. So the mixing occurs in waves that are like every 20 seconds to make up about 80% of the contractions within there is all about mixing the chyme. And then the peristaltic waves are a little less frequent, but they're much stronger than what those mixing waves are. Now, the emptying of the stomach, so the amount of time it takes, or the amount of time the food remains in the stomach, depends on the type of the food and the volume, so how much there is. So the X of a typical meal, just to kind of put it in perspective, is about 3 to 4 hours. And that pyloric sphincter remains partially closed due to just kind of a mild, constant, or tonic contraction. And then the peristaltic contractions of the stomach, when they're strong enough, they force small amounts of chyme through the opening called the pyloric pump. And that increased motility leads to an increase in emptying. And then hunger pangs actually are contractions due to low glucose level. So that's what kind of triggers that feeling of hunger, is the amount of glucose in the blood. So whenever it drops to a certain point, that stimulates our hunger. So regulating that emptying. So if the stomach empties too quickly, there's not enough time to digest everything. So it's reduced. It also produces very acidic chyme. There's not enough time for it to mix, so it becomes very acidic, and that can damage the first part of the small intestine, which is the duodenum. However, if it's too slow, the stomach wall actually can damage itself from all that acid. So you want just the right amount of acid mixing in there, enough to help break down the food, but not so quickly, again, that the acid damages the small intestine. So the neural mechanisms that stimulate the secretions are also what's going to increase that stomach motility. So the major stimulus for this is distension of the stomach. So again, those rugae, they expand or distend. That distension is what's the main trigger. And then increased motility will increase the emptying. There's also hormonal and neural mechanisms in the duodenum, the first part of the small intestine, that decrease gastric secretions and motility to prevent the relaxation of the pyloric sphincter. So it can also keep food in the stomach a little longer. And then the enterogastric reflex and the hormone CCK are major inhibitors of that gastric motility to decrease the rate of stomach emptying. Now the vomiting mechanism, so this is a protective mechanism against toxic or harmful substances. So again, if something is ingested, like a virus or a bacteria or something like that, that's potentially harmful, the body's defense to that is just expelling it through vomiting. So this can result from any irritation anywhere along the GI. So it's not just the stomach, it can be in the small intestine as well. And what triggers it are action potentials traveling to the vomiting center, again, found there in the medulla oblongata. The vagus nerve and spinal accessory nerves are what are going to trigger that reflex. So looking then at our small intestine, we have 3 main parts. So the duodenum, as we mentioned already, that's going to be the upper part that's connected to the stomach, then the jejunum, and then finally the ileum that's going to release into the large intestine. So the duodenum and jejunum are the major sites of absorption. So this is where most of our food and water are going to be absorbed. It starts decreasing as we get closer to the large intestine. So if we look at the small intestine histology, so we have circular folds or plicae. These are folds of the mucosa and submucosa. We also have villi, which are finger-like projections that stick out. This is going to increase the surface area and allow more things to be ingested or absorbed. And they're made up of simple columnar epithelium. Also the capillary and lymphatic vessels called lacteals are there that are going to help to absorb those nutrients. And then we have what are called microvilli. Again, these are going to extend off of those. These are made up of simple columnar cells. And again, the point here is to increase that surface area so there's more membrane for nutrients to be absorbed. And those microvilli make up what are called the brush border. And this is just looking at these. So again, these villi, these finger-like projections that stick off of that epithelial. And again, they're going to allow just an increase in the surface area. We have much more membrane here than if it was just flat to increase the absorption of all the different nutrients. And if we look at the individual cells that make up those, each of those cells have these microvilli, these finger-like projections or little projections, again, just further increasing the surface area. So the combination of the microvilli on the cells of the villi increase that surface area to allow for a larger absorption of nutrients. So if we look at some of the different types of simple columnar epithelium, so we have absorptive cells. Those are the cells that have the microvilli that we just talked about that produce digestive enzymes and absorb all of the nutrients or the food. So again, the more there are, the higher the surface area, the more absorption. Goblet cells, we mentioned these before with the respiratory system. They're producing mucus. Granular cells help protect against bacteria. And then endocrine cells make various hormones. Think about our endocrine system. So those endocrine cells produce hormones to help regulate the process. There are a couple glands here, the intestinal glands. These are tubular glands at the base of the villi, and the duodenal glands are also tubular in the submucosa that form there at the base of the intestinal glands. As we move at the top part here, so the duodenum, this curves to the left, kind of the head of the pancreas there, and the curve, it ends with another sharp bend that joins to the jejunum. We have the major and minor duodenal papillae. So we think about like the taste buds on our tongue, but here the papillae are openings to ducts from the liver or the pancreas that allow for enzymes to flow into the small intestine. Then the jejunum and ileum, so the gradual decrease in diameter as we move through the small intestine, but also a thickening of the intestinal wall, the number of folds, and the number of villi the farther away from the stomach. Again, the jejunum and ileum, we have a lot of nutrient absorption occurring here. There are what are called Peyer's patches. This is part of our lymph system, so lymphatic nodules form here in the mucosa and the submucosa that help absorb, especially fats, into our lymph system. And then we have the ileocecal junction. This is where the ileum meets the large intestine. It has a ring of smooth muscle called the ileocecal sphincter, and a one-way valve called the ileocecal valve. So that's going to prevent things from moving from the large intestine back to the small intestine. Some secretions of our small intestine. So fluid, primarily made up of water, electrolytes, and mucus gets secreted. Again, the mucus, just like we've talked about, provides that protective nature. It's secreted by those duodenal glands, the intestinal glands as well as goblet cells. Again, just protecting against those enzymes in the stomach acid. And then electrolytes and water from the epithelial help keep the chyme in aqueous solution. So it helps keep it moist, just helps it keep moving and improving the digestion, the breakdown of the food. Some of the other things that are being released are digestive enzymes. So these are going to be from those absorptive cells. Again, this is called brush border enzymes. So some of the enzymes being released here are disaccharide-ases, which are breaking down disaccharides and monosaccharides, so like lactose, maltose, sucrose are some disaccharides. So those carbohydrates are being broken down. Also peptidases, so peptide bonds that make up our proteins. So it's going to start breaking down our proteins. So the movement through the small intestine, again, mixed and slow propulsions over to short distances. So it's what's called segmental contractions that mix with those peristalsis while peristalsis propels it. So again, the segmental contraction, so over short distances, it contracts. That allows kind of a mixing to occur, and then peristalsis just keeps everything moving. So it takes about 3 to 5 hours for the chyme to move through the small intestine, so from the pyloric sphincter to the ileocecal junction. That ileocecal sphincter remains slightly contracted until those peristaltic waves get there. That causes relaxation of the sphincter to let it move, the chyme move into the large intestine. Then cecal distensions cause those local reflexes in the ileocecal valve to constrict. That prevents more chyme from leaving the small intestine into the large intestine. It also increases digestion and absorption and prevents the backflow from the large intestine back to the small. Now, some of the different accessory organs that we see, so starting first with the liver, so this is in the upper quadrant here, is the largest internal organ that we have. It's made up of various lobes, kind of like our lungs. So the right, left, caudate, and quadrate. These are the right and left lobes are separated by what's called the falciform ligament. We have the porta hepatis. This is containing various vessels and ducts that allow things to enter and exit the liver. We have the hepatic portal vein, the hepatic artery, and the nerve plexus that come into the liver, as well as lymphatic vessels and a couple of ducts that exit the liver. So if we look at the lobe here, so again, we have the right lobe and the left lobe, with the right being a little bit larger, and that falciform ligament that joins the two. We also have the quadrate lobe shown here, as well as the caudate lobe. So 4 lobes altogether that make up the liver. So the flow of bile and pancreatic secretions that occur here. So moving from the right and left hepatic duct, they're going to carry the bile, which helps with fat emulsification or the breakdown. From those lobes, they're going to combine at the hepatic duct. Then that hepatic duct combines with the cystic duct of the gallbladder. So here's our gallbladder here, and that's going to form the common bile duct. Now the bile flows then from the gallbladder through the cystic duct to the common bile duct. Or it can actually flow back up the cystic into the gallbladder. So it can go in either direction. Now from the common bile duct, it joins with the pancreatic duct, what's called the hepatopancreatic ampule. And we can see that right here. So this is that enlargement where the two ducts kind of come together. So then it empties into the duodenum, the upper part of the small intestine, and then the smooth muscle sphincter surrounds that common bile duct where it enters into that ampule. And then we have the accessory pancreatic duct that empties pancreatic secretions that have different digestive enzymes into the duodenum of the small intestine. So if we look a little bit at the histology, here's the liver. So lots of connective tissue, that forms the capsule, the outer layer. The visceral peritoneum covers that outermost surface. Then the connective tissue septa branch into the porta hepatocis, and that divides the liver into lobules. We have nerves, vessels, and ducts that flow through the septa. And then those lobules have what are called the portal triad at each quarter that have three vessels: the hepatic portal vein, the hepatic artery, and the hepatic duct. We have the central vein there in the center of the lobule, and then central veins unite to form the hepatic veins, and then that exits the liver into the inferior vena cava. Now, hepatic cords go out from those central veins and are made up of hepatocytes, and those hepatocytes take up nutrients from the portal blood. They help detoxify different enzymes or different secretions there, as well as make new compounds. The hepatic sinusoids between the cords there have hepatic phagocytic cells that are part of the immune, so they engulf or bring in material. And then we have the bile canaliculus, which is between the cells within the cords. Now, what are the functions of the liver? So one of the main functions is producing the bile. So it gets stored in the gallbladder, but it's made in the liver. So that bile helps with digesting and excretion. So it has bile salts, pigments, cholesterols, and fats. So it neutralizes and dilutes the acid, so it provides a neutralizing effect. The bile salts emulsify lipids, so helping to break down our fats, which don't like water. So this helps break them down. So most of that gets reabsorbed to be recycled. Also, secretin from the small intestine, the duodenum, stimulates that bile secretion, also increases water and bicarbonate ion content in the bile. Again, it's helping to neutralize that acid. And then the liver is also involved in storing nutrients. So it stores glycogen, which is how we store carbohydrates. Fats, various vitamins get stored in the liver. Some of our metals, like copper and iron, our different minerals get stored there. And that hepatic portal blood comes from the liver, from the small intestine. So as things are moving through the small intestine, they go straight to the liver, and then they enter into the cardiovascular system or into the lymph. So this hepatic portal vein connects the small intestine to the liver. In addition, we're processing nutrients, so various amino acids to make energy producing compounds. It transforms unusable nutrients to usable things. It helps with the formation and activation of vitamin D. It helps with detoxification. So those hepatocytes remove ammonia. As proteins are broken down, that amino group produces ammonia and then urea. So it helps detoxify it. Those phagocytic cells get rid of worn out blood cells as well as bacteria and debris. And various new molecules get made. So like albumin, some globulin, some clotting factors are made by the liver, as well as cholesterol gets synthesized by the liver. Now the gallbladder, this is that kind of green-like structure, again, underneath the liver, but it stores the bile. So the liver makes it, gallbladder stores it. It also has those rugae like the stomach does, so it can increase in size, have the inner muscularis and then the outer serosa. The bile arriving constantly from the liver gets stored and concentrated there at the gallbladder. It's stimulated by that CCK that we mentioned in the intestines and vagal stimulation for production. And then the bile exits through that cystic duct into the common bile duct that then goes into the duodenum. Gallstones can occur here that's just precipitated, just meaning it's more solidified instead of in solution. So precipitated cholesterol occurs because, again, the bile is helping to break down our fats, including cholesterol that come from the liver. So those gallstones, known as the cholesterol comes out of solution, can block the cystic duct, and can also occur because of drastic dieting that occurs and it concentrates that cholesterol. So if we look then at the pancreas, so the pancreas is both endocrine, meaning it has hormones that are made, as well as exocrine because it's secreting various enzymes. It has three parts: the head, the body, and the tail. Again, the endocrine portion, those are the pancreatic islets or the islets of Langerhans that make insulin, glucose, and somatostatin. Again, we have exocrine functions that are releasing things, so groups of acini, those grape-like structures that form the lobules. Those secretion from those acini go into the intercalated ducts, into the intralobular and then interlobular ducts, into finally the pancreatic ducts that go and join with that hepatopancreatic ampule we just talked about with the liver. And that's controlled by that sphincter that allows emptying into the duodenum. And then just looking again, we have the three parts. We have the head, the body, and the tail. Here's that common bile duct coming in there. We have our endocrine, so those pancreatic islets that are releasing those hormones like insulin that are coming in there, as well as the acini that are releasing the digestive enzymes. Various secretions here of the pancreas that form what are called the pancreatic juice. Again, aqueous, so it has water as well as enzymes that are there. The aqueous portion that's made by the columnar epithelial. We have various ions like sodium, potassium, and bicarbonate that help with that neutralization, as well as water. And then various enzymes that get made here. So trypsinogen, that gets activated into trypsin, which helps break down protein. Chymotrypsinogen, that gets activated by trypsin to make chymotrypsin, another enzyme that breaks down protein. There's procarboxypeptidase, that gets activated to form carboxypeptidase, also helps with the breakdown of proteins. We have pancreatic amylase that helps with the breakdown of starch, pancreatic lipase that helps with the breakdown of lipids, as well as deoxyribonucleases and ribonucleases that break down DNA and RNA. So the pancreas releases enzymes to break down all of our molecules. So our nucleic acids, proteins, lipids, and our starches or carbohydrates. Various enzymes from the pancreas help break all of those down. Now those secretions, here we have interactions of the duodenal and pancreatic enzymes. So again, the small intestine releases enzymes as well. So enterokinase from the small intestine, the duodenal mucosa attached to that brush border, that activates that trypsinogen that came from the pancreas into the active form, which is trypsin. That then activates chymotrypsin, the trypsinogen that makes chymotrypsin. Trypsin also activates the procarboxypeptidase to form carboxypeptidase. So a lot of these are made in an inactive form, hanging out, waiting to be activated. So it's kind of like a domino effect. Once one enzyme gets made, it activates another. So these enzymes like trypsin and chymotrypsin are made in an inactive form and then converted to the active form. So they're referred to as proteolytic. Again, they're breaking down proteins. As I mentioned before, the amylase breaks down the starch. So there's salivary amylase in our mouth, pancreatic amylase that gets released into the small intestine. Same thing with the lipase. There's one in the saliva, also one from the pancreas to break down lipids. And then those nucleases are digesting DNA and RNA. If we look a little bit closer at the bicarbonate ion production here, so the enzyme carbonic anhydrase gets made by those epithelial cells. That forms carbonic acid. And as we talked about with the respiratory system, carbonic acid breaks down to form hydrogen ions. So it's an acid, it releases hydrogen ions that decrease the pH, as well as bicarbonate. So the hydrogen ions are exchanged for sodium, which are then removed by the sodium potassium pump which needs energy. So it's an ATPase that moves sodium out, brings potassium into the cell. So it creates this sodium gradient to where there's more on the outside than there is on the inside. That's important to provide energy for lots of bringing in other nutrients against their gradient. Now that bicarbonate are then transported into the intercalated duct and exchanged for chloride ions. And this results in the pancreatic juice having a lot of bicarbonate. Again, that helps neutralize the acid coming in from the stomach. So then the regulation of those secretions, again, parasympathetic, rest and digest. So the vagus nerve, that cranial nerve, causes the release of those enzymatic juices or the pancreatic juice. Secretin, the hormone from the small intestine, from the duodenum, causes the release of those pancreatic juices that have a lot of bicarbonate, again, helping to neutralize the acid. And then CCK from the small intestine also stimulates the release of those pancreatic juices.